listening to the Cancer Assist podcast, hosted by Dr. Bill Evans and brought to you by the Cancer Assistance Program. Wherever you are in your experience, we're here to provide help and hope as you navigate cancer prevention, treatment, and care. Help when you really need it.
Welcome to the Cancer Assist Show with your host, Dr. Bill Evans. That would be me. And today I'm talking to Owen Roberts, who's the Chief Executive Officer of the Center for Probe Development and Commercialization. Welcome, Owen.
Welcome. Thank you for having me. And that's quite a mouthful, that name, isn't it?
It is a long name, and we're going to go into it a bit in terms of explaining to our listenership what that all entails. And it's quite an interesting story for Hamilton and McMaster University. Before we start, I always say a little bit about the Cancer Assistance Program for our listeners, in case you haven't heard it before. It's a charity in Hamilton that provides a variety of free services for cancer patients, including free rides to and from the cancer center or other medical connections with doctors and hospitals and what have you. It also provides equipment loans, wheelchairs, ambulators, other pieces of equipment to help to keep patients safe in their homes, nutritional supports, urinary incontinence supplies, amongst other things. So it's practical aids to cancer patients. And one of the things that the Cancer Assistance Program supports is this podcast, and in the hopes that by making people, the general public, aware, they feel more hopeful about cancer, and helping patients as well with their particular journey by giving them more information about their illness and how they can be supported in their cancer journeys. So that's the Cancer Assistance Program. Great charity here in Hamilton. It's amazing to me that there aren't similar cancer assistance programs elsewhere in our province and indeed across our listenership, which now goes to 5 continents. I'm amazed that there are people out there listening to us. So this will be an interesting program for me, and a bit of a different podcast for me, because we usually talk about cancers, various parts of the body, the support services that are available to patients in our community. But this is about bringing new diagnostics and therapeutics to cancer patients, and really capitalizing, I think, correct me, Owen, I'm going to let you talk in a minute, from the bright minds in universities and commercializing those ideas and getting them into the marketplace, something that I don't think universities have done particularly well in the past. But before we go there, why don't you tell us a bit about yourself? How did you get to where you are as the CEO of this company?
Sure. Well, first off, though, I'd like to congratulate you on this great program. I mean, we've all been involved with cancer in some form or another throughout our lives, and it's a terrifying experience. And the help and the education that you get along the way is so important to making this tough part of your life a little bit more bearable, and it's very important for the family. So first off, congratulations for what you do. It's very important, Sarah. My little life and how I ended up, as I said to you earlier, Sarah, I'm just a finance person who's gone wrong, took a wrong turn about 25 years ago and kept on going from there. I actually, as I came through, I was in structured finance for large U.S. institutions in both New York here and here up in Toronto. And that was my background. And just around the 1999 period, I've been doing it for about 10 years. And a good friend of mine came up to me and said, I had this idea to start up a biotech company. Would you be interested in starting up a biotech? And you're kind of one of those forks in the road of your life where I'm either going to be doing this and I got to fully commit to it, or is there something else I want to give a try for? So, it was 1999. It was the biotech bubble, and everyone was making a lot of money. It seemed like a great way to make a lot of money real fast and then retire on my boat and never be seen again. And of course, I jumped in with both feet. It was and the biotech bubble burst, but that was okay. I decided to stay around because, frankly, I really enjoyed my previous career in finance. I was very lucky with a lot of people in the areas I worked, great teams. But the one thing about biotech, medical research, etc., if you're a curious person, it's almost an unending exploration of new developments.
Indeed.
When I started in 2000, it was the Genome Project. The Human Genome Project was just coming to an end. And in fact, this is in large part a direct line from that Human Genome Project to this today. I'm talking to some of my colleagues about what it is we do now and how we target some very specific proteins, et cetera, with either probes or therapeutics. We didn't know when I started, and I like to think I'm not that old, but 25 years ago, we didn't know all the proteins. We didn't know the functionality. It's an amazing amount of knowledge that's come through. Being a finance person, trying to learn, why is this protein called ACVR1 here but L2 there? And it's like, well, before the Human Genome Project, we didn't know they were the same protein. The same gene came from it. So we gave it two different names because we thought it did two different things. Well, protein in this situation did one thing, and the protein over here, the same protein did something else. We didn't realize it was the same thing. And, you're driving blind in a lot of this, and it's really expanded what you can do. So anyways,
it sounds like a steep learning curve from finance to this biotechnology.
Unbelievably lucky with some very patient people with me who held my hand and took the time to explain to me what it was that was going on, and kind of, as I used to say, dumbing it down for the finance guy. And but slowly, after 25 years, I should have learned something. I should have learned something by this. That's actually, my friends who knew me from the finance go, I can't believe you know this. Is like 25 years. I probably should know a lot more, but we'll go from there. So, so started up a company back in 2000. It was focused on antibiotics. We sold it in 2014. Formed another company in 2014. Also in anti-infectives, and then also got involved in a couple other projects. Investors I knew, lawyers I knew from, they had this view, you succeeded once, you must be able to succeed again. And, actually I've had, a fair bit of luck and success, and worked on some interesting programs. And the one company that we had formed was just winding down because we were going to sell the assets again. And a colleague of mine, actually one of my old board members, came up to me and said, "Oh, and do you have time? We're restructuring the Center for Probe Development and Commercialization with a spin out of our manufacturing. Would you like to just help us do this process, help them complete the process, look at the legal agreements, make sure that it was all done correctly?" And they then asked, well, would you be interested in, restructuring and reforming and creating what is basically I like to call CPDC 3.0, which is our third iteration of CPDC. But I'll get into that in just a few more moments.
Okay.
So that's how I got involved in it.
Fascinating journey, not a straight line, but a very interesting.
It's biotech. There are very few straight lines in biotech. There are very few straight lines.
But one of the challenges in universities, a lot of smart people doing a lot of incredible things through their research, publishing in journals, but that's often as far as it goes. But until maybe a major player sees the publication and picks it up, or maybe the people working in their research labs grab the information that's come out of the university and they commercialize it. It seems to me that Canadian universities, and maybe it's more generally true around the globe, have never been very good at commercialization. But in the last, 2 decades, many have created offices to commercialize. I don't have a sense personally of how successful they've been, but it's always made sense to me that they should try and take advantage of the intellectual property that's been developed within their four walls. So this is something that's come out of McMaster. And maybe you could give us the 1.0 version of the Center for Probe and Probe Development and Commercialization.
So I will. So CPDC, I'll shorten the term to CPDC so we can keep this under to a half-hour show. But CPDC 1.0 was, and all of this that came out is the hard work and vision of Dr. John Valiant. I'm now the next person who's had the torch passed on to me but it really was from what John Valiant saw his involvement in radiopharmaceuticals, and I'm going to use radiopharmaceuticals. Radiopharmaceuticals just means where you add a radioactive isotope to a probe of some sort. I'll get into that a little bit later. That's where the probe term comes up. So you add a radioisotope to a probe, and then that radioisotope can travel to where the disease is that you're interested, and either give out a little bit of emission, like low power, so that you can see it show up in what's called a PET scan or other kinds of scanning equipment, or something that's a little bit more powerful that will actually break down the DNA and start splitting the DNA of those cancer cells, so the cancer cells can no longer function properly or divide. So that, in a big picture, is what he saw. And John, from his background in radiochemistry, he convinced McMaster. There were these Center of Excellence grants, and so he was awarded a Center of Excellence grant to do probe development, i.e. the diagnostics, the initial diagnostics, to in Canada and out of McMaster University. Radiopharmaceuticals, which is both diagnostics and therapeutics, had been around for a long time. Mm-hmm. But really, for a number of reasons, it really had not taken off. One of which I've already touched on, which is the Human Genome Project. You want to send this isotope to do its function to a very precise protein. But if you don't know the proteins, how do you know where you're sending it? So it was almost like this technology had been in the background, but it finally found a home after part of it from the Human Genome Project. Part of it, and many different things came together at the same time to make this a viable field. The nice thing about these diagnostics is the dose is so low. They are, considered very, very safe. If I'm going to give someone a dose of radioactive material that for a diagnostic, it's about the same or less of the radiation you get from being in an airplane.
Yeah.
So it's very low. It's very low. So these are very generally considered safe. But you need the equipment that could, once the radiodiagnostic is at a cancer cell, you need a machine that can now read that, and so can see where that is. And the nice thing is you can actually start to see volume of the cancer cells there as well. And it can be more precise than just looking at through an MRI or other, historical kinds of scans.
And often it's because it's picking up the function of the tumor rather than the shape and size, right?
That's exactly. So going from 3.1 again, John Valiant really looked at, as he built up from 2008, when this was first got the Center of Excellence grant from the government of Canada, and it was hosted by McMaster. They were focused on the probes and a lot of the research, et cetera, going around the probes. But at that time also, people were starting to see and understand how to make this on a therapeutic side as well. In therapeutics, you use a more powerful isotope, normally a metal, and that has enough energy to break up the DNA. The diagnostics, all they do is glow. It gives a little bit of a glow for a machine to pick up and a camera to pick up, and that's it. But the therapeutics has to be able to break up the DNA. And that field was just starting to expand because the knowledge about where it's going, inside the body, how it was being excreted outside the body, how you could measure the amount of radiation, all these technologies, all these understandings were being generated
together.
Exactly. Right. So around 2014, John spun out the research side from CPDC and created Fusion Pharmaceuticals. And Fusion Pharmaceuticals grew from 2014. It became a public company, and I should have had my dates down, but I think it was 2016, maybe 2018. Someone's going to slap me around if I got that wrong, but it went public on the NASDAQ around that time, and continue to grow and focus on the therapeutic side. The diagnostic side kind of stayed behind. We focused at CBDC and what I'll call CBDC 2.0, which was a little bit more on the manufacturing distribution across Canada of these diagnostics, helping set up sites across Canada. You have to remember, so for a diagnostic, say you've traveled down to Toronto or down to Hamilton or somewhere and you're told you're going to get a scan for to check for potential cancer. You're going to have your scan at 9:00. Well, the people who start preparing that dose that you're going to take, and it's going to be injected into your vein, start working on that about 2:00 in the morning of that day, and they prepare the what we'll call the probe or the cold. The probe simply is that little segment of chemical matter that gets attracted to the protein in the cancer. So the probe travels there. Sometimes they call them ligands. Sometimes they call them probes. I wish they'd just pick one, so people don't get confused. But we'll call them probes because it's in the name of our company. So I'll stay with probes. So that probe goes to a protein, and you just attach. Normally for a diagnostic, it's something like a fluorine-18 or a gallium-68, which are less powerful isotopes. So they make up the probe, and then at the end they attach the isotope, be it gallium or fluorine. And so this is now about 6:00 in the morning, and so now they have to do the QA/QC, make sure it's all ready to go and everything worked right. They have enough radioactivity in the dose so that it can be shipped off to the hospital, and you got just a few hours to get to the hospital, and it shows up at 7:00, and you have the patients all lined up ready to go, and you give the patients their dose, because for these isotopes, their half-life is about 100 minutes. So it's very short. So you're, get it in, light it up, because, the person you dose at 8 a.m., you give X amount, but by 11 or noon, you got to give them more to get the dose up. And then by, the end of the day, it's all washed out anyway. So this is just-in-time medicine. So for the diagnostics, you got to bring the people in. If you have anything go wrong along that pathway, you might not be able to dose the patient. And you have some person who's being told that they might have cancer, and that they have been brought in, and this is the first step in how to figure out how to treat, how severe the treatment, how severe your cancer is. And if you have any kind of failure along the lines, you have unfortunately a failure, and you have someone has to go up to a patient and say, "We're very sorry, it's not today, it's being done," and we have to now figure out where you fit back in the queue, and it's very stressful. So being able to produce consistently and not have failures is hard, and it's a just-in-time medicine, and that's something that CBDC and CBDC 2.0 was working very hard on. We helped some other sites across Canada also get up to being consistent on delivery. We had a very nice joint venture with UHN called CanProbe, and we were doing probe trials with UHN. But these are very, it's very stressful to be able to make these in a timely manner.
Now, the radio-labeled isotope, where is that made? Is it made in a nuclear reactor somewhere on campus at McMaster, or where is
Sure.
a radio-labeled fluorine or gallium?
So I'm going to generalize so that we can keep this under 2 days. I guess no, but I'll to be in general. So as I mentioned, when you're doing a diagnostic, the isotope is a less powerful one. So it takes less energy to create it. So most diagnostic isotopes are either made on a cyclotron. The cyclotron basically speeds up neutrons and spins, and then they hit a target. In the case of fluorine, the target's oxygen. And so you knock off, you destabilize the oxygen, becomes fluorine-18. Another one that is done is gallium-68, those ones, that one can be done again in cyclotron, or they can actually have desktop generators that, slowly generate enough of this. So those ones, less energy, done in smaller, places or in sites. On the therapeutic side, I know we're jumping around a little bit, but to answer your question, that's a more powerful, isotope. Because you want it to do something more than just,
it's going to break up the DNA.
It's got to break. The DNA actually doesn't, they don't actually take that much to kind of split up, but you have to be focused. It has to be there. So those isotopes are often can be made at a nuclear reactor, can come from actually reprocessing nuclear waste. Canadian Nuclear Laboratories has a good supply of that. But those take much more energy, and those are generally a metal of some form. So you're talking about actinium, isotopes like that. So they're they take more energy. Lutetium, which can be made at McMaster, they take the target, the original metal, and they would put it in a nuclear reactor, and the nuclear reactor bombards it for a fairly long period of time, and then pull it out. Then you have to, some of them are lutetium, some of them are other things, so you have to purify your lutetium in that case, and then you can deliver it. But the nice part about that is the half-life for that is days, much longer. So in the case of something like lutetium, it might be they might be produced, and it is produced at McMaster. It could be produced at Bruce Power, just on the shores of Lake Huron. And they have Bruce Power has a very good program that they just started. They produce lutetium, but there's enough time to get shipped, either to, I can't remember if it's at South Carolina or in Germany, to be refined and purified. And then it's actually shipped back in many cases, right back to here to Hamilton to get processed into the medical product. And even then, it still has days that it can be shipped because the half-life is so
much longer.
Yeah. So you have time. It's not infinite time. It's not like the medicines that you would see at a pharmacy, which are on the shelf for months, years, etc. But they will last for a much longer period of time.
So some of our listeners would be familiar with PET scanning. So
yes, yeah.
And the situation there is just you're radio labeling glucose with radioactive fluorine, like your F18. And then the cancer cells are taking it up because they tend to metabolize glucose more rapidly. So then they can be under a scanner and see the hot spots, which are the areas of cancer. And so staging. Here you're using different substrates to go in, which are part of the targeting. So you get very specific interaction. And you're particularly working on one for prostate cancer.
That's right.
Can you talk about that a bit?
Sure. Well, I'll even step back just 1 second, because what you want is, in the case of the glucose, this is actually called FDG-F18. So this is the most common radio probe, radio diagnostic out there is FDG. It's generic, so it's cheap. It's fairly simple to make. It's a sugar with, as you said, an F18 attached to it, which is fairly simple to do. Said by the finance guy who's never put his hands under a hood, so I can say it's very easy to do. I have no clue, but that's what I get told. So FDG hits a lot of different cancers. Also, as you mentioned, though, it is basically a sugar, so it's showing where the glucose is being taken up, and as you said, relative to other cells, the cancer cells, which are growing rapidly, need to take up a lot of these sugars. So relatively too. So what you want to get is the biggest difference between a normal cell and the cancer cell, because then you can, separate them on the screen. The question has become, I like to think of FDG as the first generation. I know there's a lot of academics who would say, no, 1960s, we were doing this stuff. No, but the real medical utility in PET scans, FDG. It's, again, ubiquitous out there. But now they've said, okay, well, we're trying to get the difference between the cancer cell and the background. the normal cells. So let's start looking at, again, back to the Human Genome Program, what kind of proteins might be specifically overproduced by a cancer in a cancer cell. So as the cancer cells grow and proliferate, do they have one specific, can I find some protein that's unique to a cancer cell that's not really out there in the regular healthy cells, so I can differentiate between the two? And PSA, many men, I think, have had their PSA levels checked. That's for prostate cancer.
Prostate-specific antigen?
Antigen, yeah, exactly. Membrane antigen, okay. But, and actually the membrane is important because it's on the outside. And that's
the PMSA.
Yeah, exactly. PSMA, you're absolutely right. But that's why we're so interested in the membrane, because it's more specific to prostate cancer than anything else. Now, your listeners and the other researchers will say, well, actually, this cancer also seems to produce in that. But for prostate cancer, it is quite large. So if you know that, which is, again, something we barely knew 25 years ago, well, then all I need to do is find a probe that binds to that membrane, the surface membrane with PSMA, that binds to it. And if it binds to it, and I put a weak little fluorine-18 or gallium-68 attached to it, well, now I've got it radio labeled.
Right.
I've taken an isotope and I've attached it to there, and I can see how dense it is and by how much it glows. I can actually get an idea of volume and et cetera. And even more important, if the disease is metastasized, i.e. spread throughout the body, and stage 4 cancers, they generally metastasize, well, this becomes a big problem for something like prostate cancer or other cancers. Where has it gone in the body? And because now, it's normally, if you have prostate cancer, they're looking at the prostate bed in the groin, and they may not know where it's spread throughout the body. But with these radiodiagnostics, you inject it, and it will go wherever the PSMA is. And you can now see, you've metastasized, and it's here, it's here, it's here. We have to make sure we biopsy here, here, and here. And the other thing is, that's the diagnostic side. And that's so we are involved with a clinical trial for a diagnostic called PSMA 1007. And there are a couple PSMA diagnostics out there. Each has its different characteristics. Each has its different utility. So, and I'm not here to go on, but promote, but also it would take at different stages of the disease, different diagnostics will have different utility. And so they're all important tools to have. And the more tools in the toolbox, the better.
And particularly, I'm just thinking of an individual I know who, has had treatment for his prostate cancer, and his PSA was normal for a protracted period of time, and then it started to rise. But other imaging, the anatomical imaging we can do with a CT scan or an MRI, didn't show anything. And so they didn't know really where it was coming from. They ended up providing radiotherapy to the prostate bed, so to speak, the area where the prostate was. It was kind of like shooting in the dark, whereas what you're describing is a way of actually finding where the PSA was coming from, because if it's got PSMA, namely the membrane antigen, and there's enough of it, you can find the location. And maybe you should be shooting with your radiotherapy to a different site, or using a systemic therapy to manage the disease. So you can see how this is a very valuable diagnostic tool.
Well, and actually we have, obviously gone to the next step is, take the same probe, attach a more powerful radio isotope to it.
And now you have a therapeutic.
Now I won't get into all the chemistry in between, but that's basically it. You take a more powerful, some people like to say, we're attaching a little cellular bomb to the probe, so that I don't like the term bomb because it sounds more violent than it actually is. Really, if you step back, so many of us have had know people who've had radiation treatment, where they try and blast the cancer with radiation, which does the exact same thing. You give it enough energy that the DNA is no longer lined up as functional, etc., and so it can't reproduce because the DNA is no longer functional.
But the external radiation's got to go through a lot of normal tissue, whereas your radiopharmaceutical is just right in proximity, right up close to it. So
that's the idea, and we've gotten, some of the biggest advancements in the last couple of years has been how to, understand the dose of the radiation that's being given, how to find the isotope that matches up nicely to how long the probe's going to be there, so you don't have other isotopes floating around in the body, et cetera. And so you're trying to manage the dose of the radiation, and it's, it sounds easy, but it's a lot of it's quite complex. But you're absolutely right. So the Health Canada just recently authorized the first radiotherapeutic for disease in Canada. And surprise, surprise, it's for prostate cancer, and it is in fact, targets PSMA, and it's called Pluvicto. And it's not a product I have any say in. Novartis has it, and it may become available and marketed in Canada shortly. There's as anyone who's been in pharmaceuticals or biotech knows, just getting it registered and getting Health Canada approval. That's step one. Then you have to go, okay, how do you train the doctors to understand this? How do you get, compensated for this? How do you get, this? And also there's a whole, you don't snap your fingers the next day and everyone suddenly knows, oh, I know how to dose people with this. I know the perfect patient for this. We got a long way to go. And, for right now, it was only, I believe it was only registered for people who have stage 4 metastatic cancer, so spread throughout the body. And you can see how valuable that would be, because you will go find where the disease is in the body, come back. But you have to remember, people who are at stage 4 are very sick, and so it's tough. But that's generally the path for new drugs. It's always given first to the sickest, unfortunately, as we continue to understand, and then everyone Health Canada and the FDA, as frustrating as they can be, but they're frustrating because they're there because their first job is safety.
Yeah, gotta protect.
And so they work that way for safety. So no matter, you can, as a drug developer, you can say, no, I wish I did, but at the end of the day,
it's the right thing.
Well, as a FDA person once told me, the safest thing I can do is not approve a drug, because no one will ever yell. But if I approve a drug and someone gets sick, I can end up in front of a committee and getting yelled at. It's like, so if I want to go home at the end of the day, I get paid the same whether I approve the drug or not. So it's actually in my incentive not to approve any drugs. I should, and then I could, never be bugged. So they do a good job. You're not going to get any complaints about either the FDA or Health Canada from me.
It's interesting to think, though, how this may impact who even delivers cancer therapies in the future, because typically it's nuclear medicine people who understand a lot of the dosing and delivered radioisotopes to patients for diagnostic purposes for eons, right? And at one point they did a lot of the diagnostics, because when I was a young oncologist, you ordered radionuclide brain scans, liver scans, bone scans. And then along came CT, then MRI, and we order a lot fewer of the nuclear medicine scans because they're a little less clear, shall we say, in defining the anatomy. And I think in some respects, nuclear medicine kind of went into quasi-dormancy, and it wasn't that interesting a career. But this is going to change the dynamics. I know when PET came along, there was a great resurgence of interest by nuclear medicine physicians. And as more and more of these targeted kinds of radiodiagnostics and radiopharmaceuticals come in, then they're going to become an increasingly important player in the treatment of cancer patients, it strikes me. So we're in an interesting phase right now, I would say.
I would completely agree. And I think you're hinting or going along the exact same path. it's always several things come together at the same time. We had to have the Human Genome Project, blah, 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 but the technology about the PET scan, the clinicians who knew how to use it, et cetera, and all these pieces coming together at the same time, and all of a sudden you take a big leap forward. And then you go along, and then there's, 5 years later, someone comes up with something else. So, what you're seeing here is what happens when several important steps come together at the same time. The utility of radiodiagnostics took a couple things. I've talked about the Human Genome Project before, so that we at least knew the proteins that we were specifically targeting. But this PET scan technology, all of a sudden, it's a step increase from MRI or CT scans. No one's going to, move from MRI to PET if it's only at the margin something better. But when it becomes evident that, wow, I can see the volume, I can see the size a little bit more, I can
and then
measure.
You can wed the technologies because you have PET CT, so now you see the function of the tumor and you see the anatomical location at the same time. That's beautiful.
You think of the utility that gives you as a clinician, as a doctor. So those technological steps, that's what brings it together. And then you had people like Dr. John Valiant, who had been interested in this space for a very long period of time, and all of a sudden, and it's not just him, but others, then their technology takes off. And we're getting right now more and more researchers, because the field is taking off. It's very interesting. And we do a great job here in Canada. On the research side in this space, we do a lot. And it's only recently now that we've been recognized, which is quite nice. Both here in Hamilton at McMaster, which has a research facility, in McGill, and in the Quebec corridor of Sherbrooke, et cetera, they have quite a good radio pharmaceutical space and research. And of course, there's the Triumph research facilities out by UBC in Vancouver. So, and there's a great deal of great work being done there. So we've been very lucky. And only recently, it's quite interesting, the rest of the world has recognized it long before, in our typical Canadian fashion. We don't even realize it. But in the last 6 months, there's a small company that called Artemis out of Vancouver that has gallium generators, just got bought for $90 million US. Point Bio, which one of the previous CEOs of CBDC started up here in Canada, just got bought by Lilly for $1.4 billion US. And then Fusion Pharmaceuticals just got bought by AstraZeneca for $2.4 billion, which is, the rest of the world is seeing the value that's here. When we went from CBDC 2.0 to CBDC 3.0 was when we spun out our manufacturing into a company called Atom B, which is still located at McMaster, but is building a large facility up by the Hamilton Airport. And it attracted, more than $90 million Canadian to build the facilities there. But they they manufacture the radiotherapies, i.e. those the probes with the longer lived isotopes for all of North America. And they do a fantastic job, and they have contracts with, all the major pharmaceuticals. They're very busy. So
I think a lot of Canadians are totally unaware of this success story. It's got well-hidden success story.
3 of those companies are within,
a few miles of us.
100 kilometers of where we're sitting. And that's, $4 billion Canadian of value that people have said, we want to buy these. The rest of the world comes in and sees it.
Well, let's take a brief break here, and we'll come back. I want to talk more about sort of the process of commercialization, going from the university research through to being traded on NASDAQ or whatever. So we'll be right back in a moment.
We'd like to take a moment to thank our generous supporters, the Hutton Family Fund and Banco Creative Studio, who make the Cancer Assist podcast possible. The Cancer Assistance Program is as busy as ever, providing essential support to patients and their families. We remain committed to providing free services for patients in our community, including transportation and equipment loans, personal care and comfort items, parking, and practical education. These services are made possible by the generosity of our donors through one-time gifts, monthly donations, third-party fundraising, corporate sponsorships, and volunteer opportunities. Visit cancerassist.ca to see how you can make a difference in the lives of cancer patients and their families.
We're talking to Owen Roberts, the Chief Executive Officer of the Center for Probe Development and Commercialization, or for short, CPDC, and really interested to hear how this has developed in Canada and spun off some really valuable companies with excellent contributions to the world of diagnostics and therapeutics in cancer. And it starts in the research labs of universities. And as we talked about a little bit in the first half, universities don't have a great history of bringing their research results to market, so to speak, and commercializing them. So maybe you could tell us a little bit of how do you go about doing that? What are the practical steps that a researcher would take in order to move from his wet lab to actually having a company that's making money?
Well, it's pretty hard. The business of biotech is a tough one. You're a clinician in yourself. The mindset that a researcher has to overcome is they can't think of themselves as the science. You can't fall in love with the science. You have to think of the clinician like yourself and say, why is this clinician, what do they want that they will then prescribe and call for this? What is the target product profile of that a clinician will want to use? And does my technology drive towards a unique product profile? So what is it? I have this wonderful bit of technology. Okay. I get, people approach me all the time and say, I have this great idea. It's great. Okay. Why is a doc going to prescribe what you're going through? What is the target product profile? Okay. The target profile is going to be once a day. Is it going to be, let's say a radiotherapeutic. Okay. What's the disease? Because remember, you're going to do a clinical trial at some time, and you're going to do this clinical trial. And if you go back to your high school science, so you're going to write out, I am going to dose a patient once every week, once every 2 weeks, once every month. What's it going to be? And what's the advantage of dosing once a week or once every 2 weeks or once every 4 weeks? Is that an advantage to a clinician? Like, does the clinician even care? Maybe they don't. Maybe they prefer to see every 4 weeks. Maybe they want to see every 2 weeks because they might want to change the dose if they're not seeing the response they want. Is there an advantage to that? Well, ask a clinician. Don't ask me. Ask a clinician. What is it they want out of this? What do they need from this? And does your science apply to that? And then once you know, okay, this is going to be the dosing. I'm going to assume because we're doing radiotherapeutics, it's going to be IV. But does it have to cross the blood-brain barrier? Do you want it to cross the blood-brain barrier? You probably don't want it to cross the blood-brain barrier because if you're doing a therapy, why do you want to have anything floating around the brain if you're trying to do, ovarian cancer? Say that's your target. I'm going to do ovarian cancer. Well, if it doesn't cross the blood-brain barrier, that's a benefit. Okay, so how's it going to be excreted? Well, I don't want, if I'm doing ovarian cancer, I probably don't want it being excreted out of the body through the kidneys, because then I'm getting, glowing around the kidneys and stuff, and I can't really see what's happening around the ovaries. I'd rather have it excreted out of the liver, perhaps. So, okay, so what do we have? We're up to something that we want dosed once every 4 weeks, because that's what the clinician wants. We don't want to excrete it out of the kidneys because we're going after ovarian cancer. For a safety profile, we want this, this, and this, and maybe we don't want it crossing the blood-brain barrier and going to the brain, because that's one less major organ that we don't have to worry about what the side effects are. And you start thinking about it. And you say, okay, this is my product profile. Find a clinician and say, do you like this? If I created something, because I think I have a technology that will make this, would you prescribe it? Would you find some utility to it? And you'll be surprised how often the docs will go, no, I have something else that's pretty much just the same. And so your technology may be, I have a different isotope that has some kind of like slightly unique characteristic, but to the clinician, they're like, Yeah, that maybe at the margin that would help. But really what I want is this. So my comment to people in academia who are looking at biotech, talk to the clinician. You're forgetting who your customer is. You think you know your customer because you think you know biology. And you think my customer is biology. And that biology, I got this thing that does this biologically, or but the customer is the clinician, the person who's going to prescribe this. Sometimes the thing that you actually invent is a way to get a patient out of a hospital quicker, or something they can take at home, which is not this case. This is not a whole medication business. But these are all things that you'll be surprised why, if you don't talk to the clinicians, you may be surprised of why they chose something. It can sometimes be, well, this would be great, but to monitor it, I need this kind of equipment, and this kind of equipment is unavailable in my hospital or in Canada, or it's so unique that it'd be too expensive to make it practical. Many decisions that you don't think of. So to your question, why do we sometimes academics fail? And it's not at all just a Canadian disease. I have this conversation all the time. Someone is they are brilliant science, and you couldn't complain about the science. It's just fantastic. But you're like, what's your product? What is it? You're forgetting your customer.
It's all about the customer. I might add, you talked about how the customer is the physician. That's a key customer, but I would say the patient is also the customer. But the things that resonate with the patient may be the 4 weeks as opposed to the weekly treatment or something, for sure. And then even the payers, because the payers are so conscious these days of what everything costs, which is often egregious in terms of its amount. So the things that make or keep people out of hospital or get them discharged sooner or make travel to and from a hospital or other healthcare facility easier, all those things become factors. So it's multifaceted, but knowing kind of the audience of sorts.
I think this is one of the things from the audience who are often, in this case, very much the patient. Remember that your doctor and your clinician is often the gatekeeper for the medications that get moved ahead.
Absolutely. Yeah.
And, we always refer to them as knowledge leaders or whatever, but there are champions in the space. They tend to be, the younger guns who are really keen, and, the two of us are exceptions to that, of course. But anyways, who are very keen, but they're the ones who will promote and put the energy in. Like, hey, this is a good. We need to scream.
So it's a very important first step, right? To know that you've got something that prescribers would actually want and need. Definitely excited by what you're doing, and I'm also very impressed at both the excitement you exude when you talk about this, because you're obviously very, very engaged in it, and by your, I would say, almost transformation from finance guy to biologist. Interesting merger of ideas and learnings and so on. I think it's been really interesting to hear from you how this center has made such an impact already, and I anticipate more impacts to come in the future. And I just really want to thank you for this podcast today.
Well, thank you very much for letting me join you. Thank you very much again for the work that you guys do. It's really important, and anything we can do to educate people in this, very scary path that they'll have to go forward on is helpful, and so that they can learn. So hopefully I was able to add a couple bricks to that whole pathway.
Oh, yeah, it's been excellent. Really, really appreciate it, Owen. This has been a different podcast in some respects, but I think a very valuable one for our listeners to hear what's going on in this area. And I think it's a developing area they're going to hear much more about. So once again, thank you.
Thank you very much.
Thank you for listening to the Cancer Assist podcast. Find more episodes, resources, and information at cancerassist.ca, or follow the Cancer Assistance Program on Facebook, Twitter, and Instagram. Thanks for listening.